Welcome to SolidScience, a podcast that is about soil.
Well, it's not about soil, but about its ecology and what we can learn from it. And it's about science.
Well, it's not about science, but how knowledge is produced. But it is about people.
It's not about people, but about their experiences. Hi, I'm Romy, a young researcher with a passion for automation.
And I'm Carlos, two soil macroecologists working in conservation ecology.
Today we will talk about the lost art of taxonomy with Lindo and Toke Høye, two experts in the field.
We are here together with Toke and Zoe, and we will try to explore a bit how taxonomy in soils is driven. So maybe Zoe, you can introduce yourself.
So my name is Dr. Zoe Lindo, and I'm a professor in the Department of Biology at the University of Western Ontario in London, Ontario, Canada. And I guess I describe myself most broadly as an ecologist, as a soil ecologist. And I mainly focus on soil biodiversity and ecosystem function and understanding global change factors that affect soil biodiversity. But that said, within my research group, we work across a whole variety of taxonomic groups, including microbes, soil microfauna, mesofauna, macrofauna, as well as examining interactions between above ground and below ground and looking at bryophyte and vascular plant communities.
And what about you, Toke?
Yeah, hello. I am a professor at Aarhus University in Denmark. I also describe myself a little more broadly than just a soil ecologist. I often say that I'm a population biologist with a particular interest in ecological monitoring. I have some expertise in Arctic ecosystems and spent quite a lot of time in Greenland in particular, doing traditional ecological surveys and monitoring. Mostly that work is concentrated on arthropods and understanding how they respond to abiotic and biotic drivers. And lately, I've found myself in interdisciplinary teams with engineers in particular to try and develop more data-rich or automated ways of studying and particularly monitoring ecological systems with a focus particularly on arthropod communities.
Okay, so today, actually, what we're trying to overcome is the issue of this lost art of taxonomy, and to try to bridge a bit between the traditional work, I would say, that maybe Zoe does to what you, Toke, do. So, Zoe, can you tell us what a taxonomist does?
Yes. I wouldn't necessarily call myself a taxonomist, though. In fact, usually when I describe my taxonomic work, I say I do it in my spare time.
Very interesting spare time with books and everything.
Yeah, part of it is just, the academic life and you're pulled in so many different directions. So taxonomist, I guess, in my definition is somebody who works at the species level to identify organisms and places them within broader taxonomic categories. And so that might extend to basically any taxonomic level. So you can work within a genus or within a family, or you can rearrange placements, basically trying to understand evolutionary relationships among organisms based on this thing that we call taxonomy, which usually involves trying to assign Latin binomial names. Yeah. Does that?
Yeah, sure. But so I was wondering, for example, using your definition, and even if we look at funding mechanisms, no one is going to fund a taxonomist, but probably someone is going to fund someone that is working with engineers in automating something. So what are the main challenges of your spare time activities?
So, I mean, going back to how I describe myself, which is more of a biodiversity scientist, to be able to generate the data that I need to ask the scientific questions that I ask about processes that generate, maintain, alter patterns in biodiversity across space and time, I need to generate the biodiversity data. And so I would say that the challenges, particularly for soil organisms, is that we haven't really been able to move to any automated system because we just simply don't know enough about the soil biodiversity to begin with. And so I would say the most sort of automated process that we have would be just moving towards molecular tools and barcoding, which to me is very unsatisfying because my enjoyment is really looking at the organisms. I don't think I could ever go purely molecular just because it takes the fun out of it for me. I'm not interested in the data as much as I am at looking at the organisms. I like the data, don't get me wrong, I do like the data. So oftentimes we'll try and pair our traditional morphological identifications with other types of data sets, and that might include molecular data sets as well. But yeah, there's no real funding for taxonomic work, which is why I say I do it in my spare time. The actual species descriptions are not things that I write into grants, are not things that I put on my annual performance evaluation.
That's sad.
Are not things that I train my students to do either, right?
Yes.
Yeah.
So based on what you said, Zoe, where do you, Toke, see the role of automation and machine-based methods in these challenges, but also in the general tasks of taxonomy?
One of the things I do is maintain a field site in Greenland that we come back to year after year, and we try to do some long-term monitoring to understand just particularly how the arthropod communities change over time, and we tag other projects onto these core activities to better understand the processes that take place and the dynamics that may help us explain variation over the longer term. So that's really an attempt to build a system understanding with these arthropods, including like all the surface active beetles and the collembola and the specialized herbivorous insects in that system. And quite a few of the species in that system, I mean, first of all, Greenland is a remarkably species poor system compared to almost anywhere else in the world. That has some advantages. It also has some disadvantages because hardly anyone studies those invertebrate communities. So we know very little about their old ecology and their general adaptations and so forth. But they're species poor, so someone naive like me can cope with the diversity and may even be able to identify some of the more common species. So part of my motivation with doing this is understanding single species responses to environmental change. How does the phenology of that particular crab spider or this weird looking staphylinid beetle, how does that change with changing snowmelt patterns and increasing winter temperatures, that sort of thing? 80% of our samples or our specimens are probably, to someone like Zoe, trivial species that almost anyone could identify in those bulk samples because the community is so simple. But I would also really like to know what rare species are found in our samples. What new stuff is there? What stuff that we haven't maybe even seen in Greenland before because no one cared to look? And for that purpose, I'd really like to automate the process of, I often say, finding the needles in the haystack, that there's a haystack that I, as a population biologist, is really excited, or I'm really excited about because of the kind of population dynamics, phenology, that sort of thing, even morphological variation. That's actually something we tend to do some studies on as well. But I'm also interested in this separation to get to that rare stuff and maybe pass those samples on to someone like Zoe or others that can actually identify them and can confirm that, oh, indeed, we found a new weevil that has never been seen in Greenland before. That's exciting. And maybe that's like a range expansion or whatnot. So I don't know if that's at least part of the motivation I have to try and build automated systems.
So I really like both of your approaches. Zoe says it's a side activity that she has where she publishes description and books. And you also say that this is just a path for something else. So what I'm wondering in this path for something else is that what is the role of taxonomists on all of these things? So if we move, because I actually never thought about this automation in the way that you referenced it. I thought that you and others that are exploring this field were trying to build machines that could go across all these species. And for example, one thing that I had in mind was, how do you deal with undescribed species? They are not there. Yeah? So, but you refer to it as, if you implement something that is long-term, it's the way that I understood it. So if you implement something that is long-term, you go there once, you go there twice, you train your machine, and then afterwards your machine does the basic stuff, and then it can, identify at least what is not basic and then separate it for. But how do you see the role of taxonomists there? Taxonomists like Zoe that then dive into species descriptions?
Probably my view has changed over the years that I've started understanding how machine learning classifiers work and how we can get help from computers to do the work that is maybe trivial, or maybe we can't afford to put humans to. So in the beginning, when I realized, oh, this field is something I need to understand, most of my presentations would have something about not counting anymore. Like, biologists keep counting stuff, and it's trivial, but it's also quite important that we stop spending time counting, because counting is something computers do quite well, and we should do something more challenging. So in the beginning, I thought, oh, we automate because the counting part is a computer task. It's not a human task. Over the years, I think I've become more aware both of the limitations of computers or classifiers. Like, we talk about these automated systems that can put things into boxes, whether it's acoustic signals or images or whatnot. And those systems, they generally only know a certain set of boxes. Like you say, they only know what they've been taught. And if you present it something that this system hasn't seen before, it will put it in one of these boxes, because it doesn't know how to not put something in a box. And I think lately we've started also looking more outside of biology for ways in which we can understand when the computer is actually not sure, when it's trying to tell us that really this doesn't fit in one of my boxes. And I think that's a very exciting part of automation that could ideally tell us which specimens should Zoe spend her evenings on, and which ones are probably something trivial that is maybe not that exciting for a species discovery, or for what's called anomaly detection. It sounds weird, but that's really the computer vision term for identifying something that is not part of the taught domain, not one of the taught classes.
Yes, but for example, we have these global networks now where we are trying to capture diversity at the scale that finding undescribed species is really, really easy, because we are exploring environments that is not only Greenland. We are exploring, everything from the Arctic to the Antarctic, literally, with everything in the middle. So when we were setting up these systems, we immediately went to people with taxonomic knowledge, because we thought immediately that's where we can find local enough knowledge that we can mobilize. But then, Zoe, how in this more or less automated world, how do you see your side projects contributing to it in a way that, because I kind of agree with you that we are still far away from a system, from a reality where automation does a dent in the volume of work that is needed. But maybe you have a different opinion. I don't know.
Yeah, I think we're still a ways away. I mean, I can understand how machines can learn, and it's actually pretty similar to how we learn as well, right? So I've actually had a small window of opportunity over the past couple of days to process my own samples that I actually collected in 2017, which gives you an idea of how long it takes me to get to some of these things. And very much like a machine, our brains are actually very similar. Like I can tell you immediately if I haven't seen something before. I can pattern recognition, sort of got it, need it sort of approach to the organisms. And when I'm looking for particular organisms, my pattern recognition can find it very effectively. Possibly not as effectively as a well-trained machine can do it, but I think my learning process is actually maybe quicker to get up to maybe 85% of what a really well-trained machine could do, but I could do it probably quicker. And I think that's where, when I think about the automation process, for myself, I don't need it because I am my own machine learned machine. Yeah, exactly. And I think, similar to my comment about, going to molecular and just doing barcodes or something, what I actually really enjoy about my work is, again, looking at the samples and looking at the organisms. And when I pour a sample into my collection dish and I look at it under the microscope, there's all this information that it's almost intuited. I can look at this diversity of organisms and because I know some of them, I see who's there, who's not there. I have a sense of where that sample was taken, the environmental drivers. It's like, oh, this sample must have been dry. This sample must be from, conifers litter. And as I sit there and I do the process of sorting my samples and counting my organisms and identifying them, at least a morphotype initially, I gain so much insight and I reinforce that machine learning and I see those patterns that emerge. And it's that deep sort of work that I get to actually indulge in that also then feeds back into my understanding of systems, relationships among organisms, relationships with their environment. And I can start seeing those patterns that then lead to ideas and hypotheses that I can then go out and test. And that includes some evolutionary relationships. It's like, oh, these organisms just, there's something about them that I think, they're probably more closely related than we currently think. And I can't really put my finger on why, but it's that sort of, I don't know, it's almost meditative, but I feel very much like a machine doing machine learning with this pattern recognition and reinforcement and yeah. So I actually have no interest in automating the process for myself because it would take away all my fun. But I do also understand that it's taken me 20 years or more to get to this level, and we don't have time. If we want to understand patterns in biodiversity and halt biodiversity loss, particularly for highly diverse organisms like soil organisms, we don't have time to train a whole bunch of people like me over the next 20, 30 years to be able to do what I do.
Yeah, you mentioned training now. So what about the training of new students? Do you suggest a mixed approach, that they learn both machine-based methods and the traditional taxonomist methods? And what are also, in terms of studying, what are the main groups that we should focus on in terms of soil organisms? And what groups are missing? Where do we have no data?
Grades. I'm trying to think of all the groups that I can't identify, and I don't know people that can identify. I would say really you could pick any of the soil organisms, and we need to train people in them, and from a variety of approaches. Absolutely.
But do you think it's relevant? Because I would like to link these two things, because we are talking from a Western world. So from a Western perspective, I may agree with you that we have to train people to go in the field and identify their species and so on. But if we think outside of North America and Europe, if we think about Africa or some places, at least in South America, I would argue that automation brings knowledge where no knowledge exists. So, and there, for example, to train someone and maintain that training for longer, it's really, really hard, you know? So do you think it's also relevant in those types of situations?
Yeah, I agree. I agree that, you know, where there are places that need and expertise, I think any approach that we take that can understand patterns in biodiversity and help conserve biodiversity and halt biodiversity loss is the right approach. My sort of attitude towards training students in my group is that I will never push taxonomy onto a student, because to do it well, you have to be interested in doing it. You know, taking students' interests and helping them run with wherever that interest is. And if it's in, you know, developing automated technologies, go for it. If it's deep dives into taxonomy, go for it. I think any and all approaches are what we need.
Yes, I agree. Toke, so in your lab, do you already teach students machine-based methods, or kind of mix both approaches and teach both, or at least give the idea that both is possible and that they both need each other?
Absolutely, the latter, that we take different approaches for different projects. So I never identified beetles as a kid and didn't grow up with that support or that expertise prior to my biology studies, and in some ways I think that's a weakness, that I don't have that intimate species identification skill. But I often find myself attracted to supervising students that do have that. And oftentimes we get good projects going with a combination of expertise where actually the student is way better at identifying a particular taxonomic group than I am. And I fully support what Zoe is saying. We really need to work with the motivation fuel that we can find in our collaborators or students broadly. So I try to work with that motivation, even if it comes from a field that I'm no expert in, and it often works then like that. I have a student that is good at spider identification. We do a project that involves spider identification. Most of the specimens will be easy to identify. A few may require some collaborator for verification. And then we use machine learning techniques to build training data set from that sample. Actually, ever since it was actually just one particular presentation I heard some 7-8 years ago that really made this click for me, that we need to pickle all the species that we have identified and put them on a shelf, because at some point they will be the gold standard reference database for all our macroarthropod image-based identification. So we still do that. We still, have this a secret cupboard with all the spiders identified, all the beetles identified, and some flies, and we'll probably never finish this, but it's an exciting pastime activity, like you say, that we built this as an investment in the future. And I think in that sense we often cross the border between doing traditional insect identification or invertebrate identification and building one little step in the process of automation.
How do you think that we can make taxonomy relevant again? Because we are talking, I'm really enjoying this conversation because you, Zoe, you seem really close to the object that we are talking about. Toke is a bit further away from it. I would see myself probably right now on the other extreme of this spectrum, where I use data that is produced by others. So how do you think we can make this taxonomy world relevant in a way that we are not afraid of showing off your work or our work on taxonomy just because it's a side product?
Maybe I can answer by asking you a question. So is because I listened to what you said about motivation and the joy you take from looking at that diversity in your bulk sample tray. And maybe I've been misinterpreting where someone like you would find that the biggest motivation, because I somehow thought that seeing all the trivial species would somehow take away time from what you would be most interested in, that particular clade or that particular genus where you really specialize and where something is still unresolved. And if you could only just push all that other noise away and work with that particular species complex, that would be the ideal. And I guess maybe wrongly, I was assuming that if we can get that hay away and give you a few needles, then you'd be more happy. But maybe that's not the case.
Yeah, I think it's different aspects of what I do. I think I would consider myself a community ecologist or a biodiversity scientist first. And so that whole community and the relative abundance of all the different species, including the rare ones or the ones that we don't have names for, as well as the common ones, those patterns are the things I think I'm most interested in, and understanding how biodiversity changes across space and time. But within that, there are the organisms that I just get then really jazzed about and have all these additional questions. And I know that they're not well placed, or you find species that you know are not described, and again, this is why I consider the taxonomy part of what I do part of my spare time. I always have I have a whole list of taxonomic work that I would love to be able to get to, and really the last time I was able to do any type of taxonomy was my last sabbatical, because that was the only time that it actually gave me that extra time to be able to do a little bit more additional work. And so I'll probably be a much better taxonomist and a much more productive taxonomist after I retire.
Aren't you then just like Carlos, that if you got a table with the relative abundance of your species, then actually you could glean as much ecological information from that table as you could from your tray? And if I gave you that table, let's just imagine that the machine could produce that table, arrange your specimens in those broad functional groups or whatever, then that would get straight to your inner dreams or your motivation.
Yeah, absolutely. I would probably be a much more productive community ecologist with those data sets just sort of, I'll say, instantaneously being generated. Absolutely. And Yeah, when I review papers and they have a species list, I look at the species list, and I look at it as a community. I look for who's there, who's not there, the different broad taxonomic groups that are abundant or rare, and to me that's the information that tells me about these patterns in biodiversity that I see. So absolutely, yeah, producing a species by site matrix of abundance data across all of my samples in a fraction of the amount of time, and possibly with a little bit better reliability than, let's say, first year master's student or an undergrad could produce, it would be great. Those data sets would be absolutely wonderful. For me personally, I still derive insight from observing the organisms myself. And I can't really describe what information I get from that, but my brain is making connections based on morphological patterns that I see that don't just come out from a list of species, even if we can identify them. And oftentimes it's, I guess, to use a word, I would say it's about traits. And you can look in a dish and see a collection of organisms, and all of a sudden there's a trait that pops out there that tells you something about how this community is interacting with some environmental variable, for instance. And sometimes it's also just other pieces of information. It's not even the organism. So, you can pour a sample in, and all of a sudden you realize that there's all these fecal pellets, and you can then start correlating it with an organism that you didn't know much about, and all of a sudden it's telling you about the ecology of this organism and maybe processes of detritivores or something like that. So there's a lot of other information that I gain by looking at my samples than just generating the species list, even though I would be much more productive if I was just handed species lists.
Okay, so there's one question that still pops up, and you already mentioned it quite a lot. It's about the future of taxonomy. So we talked about it. We probably need both approaches, and both needs to be relevant, and it will be a way that we identify the common species based on machines, and then we will have taxonomists that describe more species. But then what would you say to people that come new to the field? And also, where do you see issues regarding the future of taxonomy? So for example, regarding funding, because if taxonomy isn't funded, we can also not sustain this field. And Toke, you mentioned that it would be that you already identified all these species and they could serve as a reference for all these machine-based methods. So would that be one step to go into this funding, to enable funding just by saying, okay, we need taxonomy to give us a baseline for all the machine learning approaches that might come in the future?
I think the taxonomists should be part of these teams. So it's quite a reach to try and understand a computer vision engineer and a mechanical robotics engineer as a naive ecologist. But I think you made a good case for expanding the interdisciplinary teams. Just the conversations we're having now, I think, is actually quite insightful for how we design these processes in the future. So I think that's maybe also a way to bring more funding to taxonomy in a machine learning enabled world, that we don't do it in parallel but do it together. I think that's maybe one way forward. I guess maybe this is not really answering your question, but one thing that I often say when we speak about our automated insect identification systems is that we hope that these systems will actually enable a lot more studies of invertebrates than are currently taking place, because the job is so overwhelming. Like, so he's saying it takes her 4 years or 6 years to process samples that she really is interested about. And I think a lot of people are simply shying away from taking those samples because they realize, oh, it's going to take me 6 years to get to them anyway. So I'm hopeful that that we can also, by building these interdisciplinary teams, can get to data that are currently not collected or samples that are currently not collected.
So if I'm a new young person just entering the field, what would you recommend to me? So I want to, because several years ago I delved into insect taxonomy in rivers, but at the time I was trained as a classical taxonomist. So everything that was given to me was really far away from the things that we are seeing today. So if I'm entering this field new, what would you recommend?
I think, well, my recommendation comes back to where your interests and your passions lie, right? Because all of this takes a lot of work. Very little of it is rewarding in any sense other than personally rewarding. And so having the passion towards whatever aspect of it is going to be the thing that's going to keep you going and drive you forward. And if that's looking at the organisms, then absolutely delve into traditional taxonomy and maybe, tag that into, working with a group that does the barcoding, or, having those well-maintained reference collections for the future. If it's more about, the technology and you love innovation in that sense, then, I mean, I think there's so much that needs to be done that really it's sort of wide open. It's where do you want to take it? And then helping provide the student with the resources to just run with it. I think one of the things that I would like to see as we move forward is taking the machine learning data and making it into an accessible database for new students, someplace where they can go that's not, the primary literature from 1942 in some obscure journal, to be able to gain taxonomic information of their organism. I think that is becoming really inaccessible for a lot of people, and it's turning them off. But if there's some place where they can go on the internet and gain the information that they want, that they're seeking, and if that's through some of these machine learning data generation mechanisms, then I think new students would just be in there, right? They would really love that. They want a place to go that's not a deep dive into
The library.
Early, yeah, obscure journals from the 1960s or something.
Yeah, I think.
Okay, do you want
Well,
to add
yeah,
something?
I think I would respond that you would probably enjoy most to find a place within a big consortium of some sort, or some big team that kind of tries to connect those dots. So one of the most rewarding collaborations that I'm part of now is such a diverse set of consists of such a diverse set of expertise. So a few years back we came up with a proposal for an image-based trap to study moths, nocturnal insects. It's basically a traditional light trap with a camera, and it sounds quite simple, but because it's simple and because it's easy to standardize, I think it's gained quite a lot of traction. And now in this group we have the graphics designer to design the dashboard for this data. We have the taxonomist actually to build an annotation tool that's sensible and makes it easy for an expert to evaluate the images that are taken with these traps. We have the mechanical engineer that updates the design so it doesn't fail under lightning conditions or rain or whatnot, snow. And we have the population ecologist that looks at the data and tries to understand patterns in the data. We have the statistical ecologists, and connect both polar and tropical regions with this simple hardware system. And I think that's just so rewarding for everyone in that group, that we can see how we come in with a contribution from each of these different disciplines. And I think similarly for any tool that would work for soil organisms, such a group would be very stimulating from all aspects, both taxonomy and other, like community ecology.
Yes, I agree. I mean, this group sounds like the perfect future group, and all the projects should be established like that, because they show a way how it should go. So with this, thanks a lot for joining. I think we learned a lot, and also it shows why we have this format, to really bring together people and to explore these new ways for the future. So yes, it was really nice meeting you.
Thank you. Thank you for having us.
Yeah, thank you.
Okay, Romy. So what have we learned today?
I have to say it confirmed the thoughts I made on myself about taxonomy. So all the experience that I had in the field, it was mainly confirmed about machine learning and the future of it. But I really found this aspect nice, that the taxonomists need to be kept because they serve, or their work serves as a reference for all the machine trainings that we need to do. So we need this to train the machines, so that the check and the true data. And also the thing that Zoe said, that this experience of exploring the organisms, seeing them, seeing a community, and your brain does something with it, I think that's something we can't replace by machines. And that's also a really important issue or aspect of the field. And then, of course, the thing Toke said at the end as well about collaborative teams, both need each other. And I think they can really, together, machine learning and taxonomists, and also other groups like statisticians and biomechanics, if they work together, we can get to something that is like really great, fast, and productive as science.
Yes, I do think that's challenging. For me, this conversation was really inspiring. I really liked it. I really enjoyed the way that Zoe described, in a very visual way for me, the interaction that she has with her samples. And I also see the point of including other disciplines. But what I think it's important not to miss here is that including other disciplines implies that we include a different vocabulary, for example, that we have to talk to different people with different backgrounds, but also it has to be interesting for them. And this is what I think Toke did, something that for me was also surprising, because when he was describing his approach, I think he actually made it interesting for the engineers to do something, and for the taxonomists and so on. And that, I think, is where the critical way of moving forward is, because if we approach this new, so we enter this field new, I would say that we have to position ourselves into this matrix of expertise, but it has to be interesting. It has to be interesting for the taxonomists to do something there, because then we will fall into this trap that Zoe was describing. Well, it's not interesting for me anymore because I don't see the organisms anymore. Yeah, I don't know. It was really.
But based on what I said, I think we can overcome this. I mean, Toke already mentioned this, the analogy that the machine finds will be the undescribed species for the taxonomists. So they use different terms, but they still describe the same phenomenon. And I think they can manage it. They will manage it.
Yes, but there's another aspect related to that I actually think we have to highlight that in the future. That is, if you have a site that you go systematically there, these machines actually can make it feasible, financially feasible to have a monitoring system, because for that particular site, unless you have a drastic change in the system, most of the diversity is known, so the machine can easily, let's put it like this, count and go through it, and in a way that it's feasible to have a monitoring system, because otherwise it's really hard to think right now, for example, economically on a monitoring system that can go over continents to sample soil biodiversity in a feasible way.
Yes, that's true. Yeah.
Okay, I think that's it. No?
That's it.